I sequence all 10 isolate I get, and most of them are Bacillus, with only 2 turn out to be other closely related bacteria. Bacillus is everywhere, on plant, in soil, water, or probably under your feet right now. They are the most diverse and useful group of bacteria that humans had ever learned to work with. We use Bacillus to grow crops, break down waste, clean water, make food and drugs. The best part is it's easy to find, easy to grow, and surprisingly forgiving to work with. So in this video, I will show you a simple yet effective workflow to isolate them. Whether you want them for your garden, your pond, your compost, or simply want to keep them as pets. To be clear, some Bacillus species can be dangerous and harmful to humans, and those are not what we're looking for, and I won't be showing you how to get them. With that out of the way, let's begin. First, we need to prepare some basic equipment and culture medium. For that, what you will need are sterilized pipettes, inoculation loop, spreader, and alcohol lamp, as well as something that can be used for heating. If you live in a relatively cold area with an average annual temperature below 10 degrees Celsius or 50 degrees Fahrenheit, you might also need an incubator. For bacteria identification, you will need a microscope. A high school level microscope is enough. Next is hydrogen peroxide. Both low concentration like this 3% you can get from pharmacy or 35% will work. You will also need grandstanding reagent, which can either be purchased as commercial kit or prepare yourself, but I recommend buying a ready-made kit for convenience. Next come with culture medium. I usually use nutrient agar. Some people also use LB agar, which works just fine. Once everything is ready, we can start isolating bacteria. Bacillus species are very common in natural environments, so we will start by isolating them from soil, the most abundant source. Soil samples usually contain a huge number of different bacteria. For a sample like this, a simple isolation method is heat treatment. Basically just boil it. Since Bacillus species are heat-resistant bacteria, heat treatment can effectively reduce the number of unwanted microorganisms. Here I add some water mixed with soil and heating it at 80 degrees Celsius or 176 degrees Fahrenheit for 10 to 15 minutes. That can eliminate most heat-sensitive bacteria while retaining the maximum number of heat-resistant ones. However, this method requires a temperature-controlled heating device. If you don't have one, simply boil it, it will also work. Next, we move on to spreading the sample onto the agar plate or using the 4-quadrant streaking method to isolate single colony. After that, it's time for incubation. To prevent the agar from drying out, I place the plate inside zip bag. They can be incubated at room temperature, about 1 to 2 days in summer, around 30 degrees Celsius or 86 Fahrenheit, and roughly 3 to 4 days in winter. If you have an incubator, you can use that as well. This is what it will look like after incubation. You can see individual colony growing on the plate. The next step is to use an inoculation loop to pick a single colony and streak on a new plate to obtain pure culture. I usually repeat this step 3 times to make sure the isolate is truly pure. The advantage of using heat treatment for isolation is its high efficiency. Most of the colony on the plate will be Bacillus species or other heat-resistant bacteria, so you don't need to spend too much time selecting colony. The downside of this method is that you might miss a lot of rare species, so it's not suitable if you want a very detailed or comprehensive isolation. However, if your goal is just simply obtain some common Bacillus species such as Bacillus subtilis, this method is very labor-saving and practical. After obtaining the pure colony, the next step is to check whether the bacteria we are isolated are Bacillus species indeed. To do this, I will use gram-stain and catalyst test. After gram-staining, we should observe gram-positive rod-shaped bacteria under the microscope, like what you're seeing on the screen right now. Since many Bacillus species are relatively large in size, a 14-time objective lens is usually sufficient, so you don't need a very high-end microscope. For the catalyst test, a small amount of fresh bacteria is transferred onto a clean surface, such as this glass slide, and a drop of hydrogen peroxide is added. Most Bacillus species should show an immediate positive reaction, which is visible bubble forming. To this point, if your sample being heat treated, show gram-positive rod on gram-staining and is catalyst positive, there's a very high chance that the isolate is Bacillus or a closely related genus. For further confirmation though, gene sequencing can be performed. I sequence all 10 isolates I get, and most of them are Bacillus, whereas only 2 turn out to be other closely related bacteria. After isolation, the bacteria are usually cultured further to increase the biomass and make it easier for downstream application. To minimize the contamination, I always start with small-scale culture, usually 5 milliliter. If more is needed later, I simply scale it up. Usually I prepare the medium in advance. Like here, each tube contains 5 milliliter of TSB medium, so when I need it, I can just grab one and use it for convenience. Simply pick a small amount of bacteria with an inoculation loop and transfer it into the medium, and it's ready for incubation. Since Bacillus is aerobic bacteria, it needs oxygen. With small culture, well, you don't have to shake the culture, but oxygen have to slowly diffuse into the medium, so the growth will be slow. If you want faster growth, then yeah, shaking it. For large culture, stirring and aeration is a must. As for the temperature, room temperature is generally sufficient. If faster growth is desired, incubation at a higher temperature can be used. For environmental isolate like this, I typically put them under around like 30 degrees Celsius or 86 Fahrenheit for 24 to 48 hours. In most cases, it's enough. If you want to prepare your own culture medium, you can start with basic protein and carbohydrate source. Here I have some protein powder from soy sauce and worm, along with some sucrose and sodium chloride. Bacillus are not picky eaters, so they can accept a wide range of food source. And if you have buffering salt, such as what I have here is dipotassium phosphate, you can add small amount to help stabilize the medium. I put my recipe on the screen. After adding 100 or 200 milliliter of tap water and sterilize it, you will have basic culture medium. However, homemade medium like this usually not clear enough, which makes it difficult to assess bacterial growth. So for small-scale culture, I prefer using a clear medium instead. This is one of the Bacillus strain I isolated. I grow it on the same plate together with a fungus. Then the fungal colony seems to hit an invisible barrier, as if something is stopping it. it cannot spread any further. Later PCR screening hints that this bacteria might be producing an antifungal compound called eturin. This might be the reason for this invisible wall. After spreading the bacteria on the agar plate, I place filter paper that has been soaked with different compound. Only the disc containing influenza name show a clear zone with no bacteria grow around it. In contrast, bacteria grow right up to the disc containing azoxystrobin and chloramphenicol, suggesting that this strain is resistant to those two compounds. Interestingly, fluenziname is actually a fungicide, yet it still has an inhibitory effect on this Bacillus strain. But on the other hand, the antibiotic chloramphenicol has no effect on it. I usually preserve those isolated Bacillus strain by mixing them with 20% glycerol and store them in the freezer. That I can keep them longer to explore their potentials and functions. So the question is, what interesting thing you might find in your Bacillus isolate?